All right, let's get a little more clarity on this protein synthesis. First of all, let's talk about the difference between DNA and RNA. So they're both nucleic acids. DNA is double-stranded. RNA is single-stranded. DNA has deoxyribose as the sugar group. RNA has ribose as the sugar group. DNA uses A to T and C to G. RNA uses A to U and still C to G. So one big difference is the nucleotide uracil is used in RNA instead of the T over here in DNA. So U instead of a T. Okay, so it's a big difference between RNA and DNA. All right, so now let's go through our transcription and translation again in a little more detail. Here's our big chromosome, right? Our big single, one big piece of DNA. And here's a gene. And here's a section of the gene that we're looking at right here. Here's the A's, T's, C's, and G's of the DNA. And in order to transcribe, this opens up and the complementary RNA nucleotides are added. And the first one that pairs with A is U, and then C to G, C to G, A to U, and so on, T to A. And we have our mRNA transcript right here. And then when it breaks off, when it's done being transcribed, it breaks off and it leaves the nucleus and it goes into the cytoplasm and it goes into a ribosome. The ribosomes are floating around the cytoplasm. They're also in what's called the rough endoplasmic reticulum. What makes it look rough is all these ribosomes are sticking on the outside edges, right? So the ribosomes take in this message, this messenger RNA, and every 3 nucleotides codes for a specific amino acid. Okay? These 3 are called a codon. And so the ribosome reads them and says UGG and goes, oh, that means I need this amino acid. And it attaches it. What's next? This and this and this. Every 3 RNA nucleotides make up a codon and they code for a specific amino acid. And this is how the ribosome builds the proper protein.